to do, but I can't do that. I need to talk about Jim. So Jim showed up in 2016 wanting to work on ticks, and he found a mosquito person because there were nobody at the time working on ticks in New Jersey. And in fact, once he actually provides MD.
Sorry.
Okay, thank you, MB, for all the support you've given Jim all these years. Thank you, appreciate it. He showed up wanting to work on ticks, and I, in an enormous feat of hubris, said yes. And I'm really glad I did, because I've learned a lot. I've learned a lot about ticks, but I also learned a lot about being a fantastic person. And Jim taught me a lot about, different kinds of intelligence, the emotional intelligence that makes people just really want to work with him and really interact with him and really bring in what we need as a community to be able to solve the New Jersey tick problem. So thank you, Jim. Take it away.
Don't go anywhere yet. So I'd like to thank my committee and everyone here for coming. And if you don't know this, Dina's hair used to be all black. And even last night, she goes like this when we're going through the talk and says, Jim, you see this white hair? It's from you. So I got you some hair color. So you can look yourself again. Sorry for the gray hair. Okay, so baseline study, thanks everyone again. Baseline studies towards a statewide tick and tick-borne pathogen surveillance in New Jersey. So I like to collect ticks, but I needed some organization. But this was not the first attempt at looking at ticks in New Jersey. Now I'm not working. Okay, all right, there we go. So Thomas J. Headley in 1940 was a professor here, wrote this pamphlet, The New Jersey Tick Problem. And he has a number of interesting insights. This building is named after Thomas J. Headley, Headley, not Headley. And we'll mention his book throughout the talk. So first, I'm going to give you a little tick biology 101. So ticks are arachnids, 8 legs, unless it's a larva, then it has 6 legs. There's 935 species, and this changes frequently. There's 193 species of soft ticks, the Argacidae, and 742 species of the hard tick. And that's not in this part of the world. We're not going to talk about it. There's only 1 species. So there's a soft tick. See, it's kind of like leathery, and it fills up with blood, male and female. This is a hard tick. Female American dog tick, male. This female will fill up with blood. They have a couple other differences I'll mention later. So they're obligate blood-feeding parasites, and the problem with that is they can vector pathogenic microbes by this method of feeding. So why are we concerned about ticks? Primarily because they cause a lot of morbidity and mortality. So this is from 2019. You can see here, where's my mouse? There it is. Lyme disease, 34,000 cases. Anaplasma ehrlichiosis, almost 8,000. Spotted fever rickettsiosis. So that name, spotted fever rickettsiosis, is a new term for what used to be called Rocky Mountain spotted fever. I'll explain that too in a few slides. Babesiosis, which is a protozoan disease, and Powassan virus. So if we look at New Jersey, Lyme disease in particular, 3,500 cases. And if you look at babesiosis, 236 cases. Compared that to the national caseload that year, New Jersey routinely has 10% of the country's caseload in some tick-borne diseases. Sometimes it's higher. And in the year 2019 in New Jersey, with the 4 Powassan cases, that was in Sussex County in about a 5-mile radius in one area, and resulted in 2 deaths. Okay, so let's look at the historic timeline of tick-borne diseases in human tick-borne diseases in the U.S. and North America. So the first one discovered was rickettsii, known as Rocky Mountain spotted fever back then. Then we move up, then some relapsing fever diseases, and then things start to heat up in the 80s. Then the big one, Lyme disease hits, okay? And then we have maybe 10 more up until 2020, newly discovered viruses. So discovered in 1906, the agent rickettsii, and we still don't have a handle on it. So let's look at some epidemiology. This is cases per million, and this blue dotted line is case fatality rate. So you see around 1950, a dramatic drop in fatality rate. That's because of antibiotics. Okay. Then you got a couple hiccups with the incidence, but the case fatality rate still decreases. But then look at this incidence. It jumps very high in the 2000s, and it's not entirely known why. But it's important to note that Rocky Mountain spotted fever is a potentially fatal disease. So this is the petechial rash, as it's called. So the rickettsial agent infects the endothelial lining of the blood vessels, compromises those blood vessels, blood leaks out, and then you get the rash. If compromised, if the vasculature is even further compromised, the vessels break down, and then the extremities become gangrenous. And Dina told me not to show that slide. But back to Thomas Headley. In that pamphlet he wrote, he said even in 1940, in the U.S., 70% of the cases were fatality in the pre-antibiotic era. In New Jersey, he said it's not as virulent, only 20% fatality rate, which is kind of interesting. Okay, so we have ticks in New Jersey. We have tick-borne diseases. The first job Dina asked me to do was make a list of New Jersey ticks. I said, I don't think there is a list. And she said, well, how many species do you think we have? I said, maybe 10, 15. She goes, well, what is it? I said, I don't know, maybe 10 or 15. She goes, okay, you need to make a list. I said, there is no list. She says, you make the list. I'm like, oh, okay. So that was my first chapter of my thesis, was identify species of ticks proven to be present in New Jersey. So the main rules were, if you claimed the tick was in New Jersey, you had to have a vouchered specimen, meaning it was in a museum and it had full collection data, where, when, and how it was caught, who caught it, or flagged it, or trapped it, or whatever, so that if someone questioned it, you can go back and look at that voucher specimen in the museum and make sure it's valid. So this is the U.S. National Tick Collection when it was in Montana at Rocky Mountain Labs. So the materials and methods was pretty straightforward. I queried all the large museums east of the Mississippi, even up to Chicago, their big museum there, Harvard, entomological collections and universities around the area. And no one had an official list of ticks that they had in their states, nor did they have anything from New Jersey. So I relied entirely on the U.S. National Tick Collection, which is in Georgia, and they provided all the data I needed. So this is the result, the annotated list of hard ticks of New Jersey. So there's 11 species. This was, I think, 1920, this was published. This is an invasive species, Hemiphysalis longicornis, discovered by Todd Rainey and Andrea Giese. And this is also an invasive species. This is found all over the world and is probably a species complex now. So that's the publication. But there are other ticks that are probably here, and we base that on what's found in nearby states. So for instance, in Connecticut, there's all these species. So we probably have Nereus and Marksii, et cetera, et cetera. But let's point out what's happened since, I know you can't see this, but let's point out what's happened since this has been published. Amblyomma maculatum is now found in Connecticut and New York, and that's established populations. The problem is Amblyomma maculatum, known as the Gulf Coast tick, transmits Rickettsia parkeri, and 50% of the populations of this tick, I think it was Staten Island, were positive for this Rickettsia. The good news is it's not fatal like Rocky Mountain spotted fever. It's just more of a febrile disease, but there is a characteristic of this disease is you get an eschar at the bite site. So other ones that are probably going to be in New Jersey: Angustus, Banksii, Marksii, and Murus. In fact, Julia Gonzalez in the back is about to publish her finding of Ixodes marksii in New Jersey. Okay, so what do we learn from all this? If you have a new state record, you have to confirm it with an expert, a tick identifier. We use Rich Robbins. I'll explain more about him later. If the specimen's in good enough condition, or if you have multiple specimens, you can isolate DNA and run a DNA barcode on it. And I'll explain more about that later. It's just basically taking a piece of DNA, sequencing the gene, and seeing if it matches same species that are in the database or similar species. And then you have to submit vouchers to a museum, or else I don't count it in my, if you know what I mean. But you also have to submit the voucher sequence to the Gen Bank to make sure that's a valid voucher as well. And we sent our ticks to the U.S. National Tick Collection and the Yale Peabody Museum. So there's also several other tick species that may already occur in New Jersey or maybe moving north. Oh, wait 1 second. So it's a standard operating procedure now that we barcode anything we find new or we don't know what it is. And we're not going to make a huge database for the public, but just for ourselves. So, okay, we got these ticks in New Jersey. What do you do with ticks? Well, any self-respecting microbiologist would crush it up and look for pathogens. So the first project I thought about was going back to Headley, the 1940 spotted fever rickettsia problem. So, whoops. There we go. Okay. And the question I asked was, is Rickettsia rickettsii present in ticks? One reason why I asked this is because I had a tremendous collection of Dermacentor variabilis, and you're not going to test those for Borrelia burgdorferi. So, you know, what would be a good idea what to do with them? And this was part of the answer. So in the East Coast, Dermacentor variabilis has historically been the de facto vector of Rickettsia rickettsii, the agent of Rocky Mountain spotted fever. And also Amblyomma americanum can be a secondary vector. In order to accomplish this, and I'm just one person, we wanted to survey the entire state of New Jersey, and we did that with the professional men and women, highly skilled, of the New Jersey Mosquito Control Association and the representatives from each county. We had a workshop. We trained them how to collect ticks and how to identify ticks, even though most of them already knew how to do it from their daily work with mosquitoes. We all went out on one day at the same time and collected ticks, and it was a lot of fun. I hope those who were involved with it. And here's some actually working in the field. We made tick sweeps for each county, and we called it the New Jersey Tick Blitz. Okay, so what do we find? So here we have Dermacentor variabilis, the American dog tick, the de facto quote unquote vector of Rickettsia rickettsii. And over here we have Amblyomma americanum. We tested 560 ticks from the Tick Blitz and 394 from my own personal collection from 2013 and 18. And we found no Rickettsia rickettsii. All we found was 1.3% Rickettsia montanensis. No, yes, montanensis, not monocensus, montanensis. And the two methodologies were not statistically different, so we pooled the data and made it one big batch of ticks, 954. So almost 1,000 ticks, no Rickettsia rickettsii in any New Jersey dog ticks. And then we also got 245 Lone Star ticks from the Tick Blitz. Again, no rickettsia. About 20% infected with Rickettsia amblyommatis, and that's considered a non-pathogen, although there's one clinical case of a febrile child bitten by a Lone Star tick, and they found evidence of Rickettsia amblyommatis in her serology, no, probably in her blood or PCR, because you can't differentiate one spotted fever rickettsia from another with serology. And I'll talk about that in a little bit. And recently, Andrea Jeezy found one Lone Star tick out of almost about 1,800 positive for Rickettsia rickettsii. Is that project ongoing? You still, yeah, and you still
Never found any more.
Oh, okay, never found any more. Okay. Okay, so here's the resulting publication. New Jersey-wide survey of spotted fever group rickettsia in Dermacentor variabilis and Amblyomma americanum. And we found no evidence of Rickettsia rickettsii, like I said, and none in the Lone Star tick either. So the only way to solve or attempt to solve the question of what's causing spotted fever rickettsiosis in the country is to engage public health people, physicians, and clinicians, because I just said that, well, first of all, most cases of Rocky Mountain spotted fever or spotted fever rickettsiosis are diagnosed clinically. So you've been running around the woods, you pulled a couple ticks off you, you go to the doctor, you have a fever, and you got a rash. Okay, you have Rocky Mountain spotted fever, or let's just say spotted fever rickettsiosis from now on. They don't usually do serology, but if they do, any other spotted fever rickettsia, and I think there's 20, is going to cross-react with whatever you're carrying. So it's not specific. So we need DNA analysis, PCR, but you'd have to engage patients and physicians and clinicians. So it's going to be a tough slog. So what's going on? Is it due to a different rickettsia? Is it due to a different tick transmitting a different rickettsia? So this question brought me back to my original list of ticks in New Jersey, where I wanted to look for soft ticks in New Jersey because none had been reported. So Dina's, as her hair is turning white, says, focus, Jim, this is on hard ticks, okay? You can do the soft ticks later. So finally, I get to the soft ticks. And the question is, where is Carios kellii in New Jersey? And here's the known distribution in 1944. And oh, look, there's one in Pennsylvania. So right next to New Jersey. So this tick, the bat tick, it's a soft tick, like I mentioned, feeds almost exclusively on insectivorous bats. And these bats are often associated with human buildings and like attics and such in the unused areas of your house. Sometimes from competition, who knows what else, bat exclusion, they will come down into the living areas and they do bite people. Okay, so let me give you a little Bio 102 on soft ticks. So, and especially Carios kellii. So hard ticks, they'll feed for days, each stage, okay? Soft ticks, the only stage that feeds for days are the larvae, and they will feed for 2 to 3 weeks and stay on that host, usually a bat. In fact, it's rarely been found on anything else, except for a dog and a human. They were also bitten. So they'll stay on for 2 to 3 weeks. So if that bat migrates or finds a new shelter, it could be a new focus of Carios kellii in that area. When they molt and turns into a nymph, and that's the other difference between this and a hard tick. Could be up to 4 nymphal instars. So, but the nymphs and the adults, soft ticks, feed very quickly, maybe an hour, hour and a half, and then they go back and they hide in their spot. And they're what's called nidicolous endophiles. They will stay in the cracks and crevices of the roost and stay inside. You're not going to find them outdoors. And another difference is the female will feed multiple times. Hard ticks only feed once each stage. So they will feed multiple times and they will lay multiple egg batches, although very small batches. Hard ticks lay up to thousands of eggs. These maybe 100 or so. So soft ticks can live for years without blood, and they can remain infectious as well. In fact, there was a story published about a tick, Argus brumptus, that was kept alive for 27 years without food. Okay, so how do you search for a bat tick? I tried it on my own, it didn't work, so I had to reach out to a bat expert. Then I've never seen one of these before, this project, so I had to enlist a tick identification expert. Oh, and there's the bat expert, that's Mackenzie Hall. Here's a tick identification specialist, Rich Robbins. He taught us how to use the morphological keys, and then we also barcoded this tick. So I called Mackenzie and I said, I want to get some bat ticks. Have you ever seen any? She said, I've been doing this for 15 years. I've never seen a bat tick. I've seen mites, I've seen flies, and plenty of bat bugs. So 2 weeks later, she called me and says, Jim, I think I have bat ticks. So she sent them over. And then Rich Robbins helped me identify them. And whoops. There we go. And here's the results. We found Carios Kelly in the first time in New Jersey. We submitted voucher specimens to the Yale Peabody Museum, and we submitted the first Carios Kellyi cytochrome oxygenase 1 sequence. That's what barcoding is. You sequence the cytochrome oxygenase of the mitochondrial genome, and that is used to help identify species. We found, Mackenzie found 3 larvae in Sussex and 12 in Mercer. And this is my skin, my back of my hand, that's a dime, and that's an engorged larval Carios Kellyi. So this is the resulting publication. Now it's now officially part of New Jersey Ticks. And it was a fun project. So what do you do with ticks? Well, like I said, any self-respecting microbiologist is going to crush the damn thing up and see what's in it. So, and there's precedents for this with bat ticks. So in the Midwestern states, for instance, Iowa, 90% of the nymphs and 3.1%, 90% were infected with this unknown rickettsia, and 3.1 were infected with Borrelia johnsoni, which is a relapsing fever spirochete, radically different than a Lyme spirochete. And also in Iowa, another study, another county, 45% of the nymphs were infected with Borrelia johnsoni. And in Kansas, no Borrelia, but 66% of the nymphs and the adults were infected with the same rickettsia as up here, in addition to 2 other rickettsia. And one interesting publication came out in 2018. Kingery took blood samples of 5,000 patients suspected of having tick-borne diseases, and they did PCR in the blood, not serology. They did PCR detecting the DNA, and they found 1 patient was positive for Borrelia johnsoni. So that's indirect evidence, good evidence, I think, but indirect, that Carios Kellyi will bite humans and transmit Borrelia johnsoni. So I wanted to branch out and get more ticks. So I assembled tick kits, and these are the partners, some of the partners I sent them to. This is Vermont, New Jersey Wildlife, Ohio State, PA Bat Rescue, and Wild Thing Sanctuary. The PA Bat Rescue and Wild Thing Sanctuary, they're nonprofit bat rehabilitators, and it's nonprofit, and I always send them donations provided we have money in the account. I have to check with the boss. So how did that work in the field? Well, first of all, like I said, I sent tick kits to all the bat researchers and rescuers, vials, forceps, labels, and small plastic bags, and of course a postage page envelope, because you don't want, people are doing you a favor, you don't want to cost them any money. And then also Mackenzie and I went out and collected ticks using a CO2 trap. And my mouse works on my screen, I don't see it working. I don't know if it's working for the other people, but anyway, for us in the audience here, there's a coffee cup, styrofoam coffee cup with dry ice in it. and it poked holes in it. And it's sitting in a device that Cheng Lu gave us, Cheng Lu Wang from his entomology lab. And we put flour in it so the things can't crawl out. And this is bat guano and loaded with bat bugs. We didn't see any ticks in this house, but loaded with bat bugs. And I gave some to Cheng Lu. He's starting a column. So on the house, we did get ticks. You can see them here. And there's some kind of shenanigans going on there, and I'll explain why later. But they don't mate like that. They mate ventral to ventral, and the male uses his mouth parts. And here is my second encounter with big brown bats. I got a little braver. Well, the first time I encountered him, I almost pooped myself, and Mackenzie said, "Suck it up and get back to work." So that's what I did. So what did we find with our collections and the collections from our collaborators across the Northeast? So we got a total of 125, and we tested 112. 7 from Vermont, 58 from New York, 28 from Pennsylvania, 7 from Ohio, and 12 from New Jersey. And we also barcoded them to see if there's any differences or whatever. So and New Jersey was the voucher specimen, the first one from the first Carioskelly I would find. And New Jersey, PA, New York, and Vermont, identical to New Jersey's, the cytochrome oxygenase one was identical. Ohio had 2 haplotypes. One was radically different. Another one was only 2 base pairs different. And I got to point out that these PA samples were taken from the eastern side. So if maybe over here there's something intermediate, I don't know. I'm not going to play population geneticist. So how did we test these ticks for pathogens? We tested individual ticks for spotted fever rickettsia because based on the published data, there was a high prevalence. But for the relapsing fever Borrelia, we tested them in what we call post-processing pools. So you have an individual tick in each tube. You crush it up, isolate RNA, and then you make a pool of the RNA, but you better save some to go back for, right? Then if that's positive, you go back to the original tubes and find your positive tick. So, and it saves on resources and time. Then we sequenced the targets, several targets, I'll show you that in a second, of a subset of each group of ticks from each state to confirm. And when you get the DNA sequence of the targets, you can confirm or at least get a suggestion of what species it is. So for rickettsia, we use the GLTA citrate synthase, AMP-A out of membrane protein. And this is just the size of the PCR product that we use. Then we submit it to NCBI for a blast or comparison. And for Borrelia, we used FLA-B, 16S, and GLPQ. And that's, again, the size. Okay, so what do we find? So for Vermont, nothing, no Borrelia, no rickettsia. For New York, 36% were infected with spotted fever rickettsia. New Jersey, 41% with spotted fever rickettsia. And in Pennsylvania, 28 were spotted fever rickettsia and 21% of relapsing fever Borrelia. And that's interesting to note that 4 ticks, I think it was 4, came from one collection and another tick came from another collection. So, and then for Ohio, just spotted fever rickettsia. So when we sequenced the 2 loci for the rickettsia, they matched 100% with those first findings in Iowa, that unnamed tick, unnamed rickettsia, sorry. So just to point out the profile of what these specimens were, 90% were engorged larvae, so they had blood in them. We've got a few dead adults from Ohio and 3 live adults from New Jersey. Okay, so we wanted to know what this rickettsia was. 2 genes is not a good enough thing to identify a new species. So I saved some DNA for Dr. Dana Price, and he started the sequencing and found out, okay, this is going to be a problem because the one he sequenced, he found evidence for 2 spirochetes. I'm sorry, 2 rickettsia. So what I did was I took further ticks, some more ticks I had, and I cut the legs off, and I did qPCR on those because you don't need much material for qPCR. So I could save the rest for Dana if they were positive for rickettsia. So I think I have 2 for you. And we're going to try some whole genome sequencing to get the whole genome sequence of this rickettsia. So I'm going to digress for a minute and mention how ticks can transmit pathogens between themselves. Okay, not a host, but between themselves. So this is these are Carios kellii. Here's a larva, 6 legs, metamorphoses into a nymph, and then, like I said, there's 3 or 4 instars of nymph, and then the adult, and fed adult, and then a mated adult. So if you pass a pathogen from one tick stage to the next, that's called transstadial. What can also happen is the pathogen in the female can be passed to her developing eggs. And when those eggs hatch, they're infected. That's called transovarial transmission. So we were curious as to whether this happens. So Julia Gonzalez, the bat whisperer, says, let's try to raise larvae from these. And Sonnenschein in 1964 said, they don't feed on anything with bats. So Julia said, well, we're going to try it anyway. Julia, sorry. So here's Julia's artificial membrane. And here's the 3 live adult Carios kellii that we captured in Mercer County, New Jersey. And she successfully fed them and they engorged in her device. The interesting thing was you need to add bat guano to the feeding device. It's a phagostimulant. So remember I said the shenanigans. Well, there was some shenanigans because they eventually laid eggs, and here's the larvae crawling on the guano. So they didn't feed. I mean, 1 or 2 maybe fed, but not engorged, and they eventually died. So 2/3 of those adults, and they were female, were positive for rickettsia. I tested 6 unfed larvae, and 3 were positive for the rickettsia, suggesting transovarial transmission. And that was pretty cool. And I mentioned it to Dina. I forgot how I just mentioned it to her. She goes, what? It was very important, but I forgot, and her hair turned even whiter. Okay, so now we have evidence of transovarial transmission thanks to Julia. So what about the Borrelia that we found? So here's the analysis of the Borrelia, and ours is right here, Borrelia Carios isolate from New Jersey, and it joins with Borrelia johnsoni, the relapsing fever spirochete found in Iowa. And that was done by taking 3 sequences, concatenating them, and then running the analysis. Okay, so the summary. The first multi-state collection and pathogen analysis of Carios kellii. I mean, it's been done before, but not multi-state. The first finding of spotted fever rickettsia in Carios kellii from multiple states in the Northeast. Evidence of high transovarial transmission of spotted fever root rickettsia, and the first finding of putative Borrelia johnsoni in the Northeast. So I mentioned that Borrelia, relapsing fever Borrelia are different than Lyme spirochetes. So these are relapsing fever Borrelia in a blood smear. So you can become bacteremic, lots of bacteria in the blood with relapsing fever, and you can actually do a blood smear or a dark field stain and see the Borrelia. You can't do that with Lyme disease because the spirochete in Lyme disease is in such low numbers, low abundance in the blood. It's usually in the tissues, which is one reason why Lyme disease is hard to diagnose. This is relatively simple. And there's interesting point is that one Borrelia dutoni in Africa is strictly a human pathogen. So the humans are the reservoir. Out in this country, usually rodents and maybe bats, but we don't know that yet, are the reservoir. The other thing is, if you treat somebody with relapsing fever Borrelia, you have to be careful and watch them because the population of bacteria is so high, it's thought at least, that when they start to die, they release all kinds of things that wreak havoc on the immune system, and it's called Jarisch-Herxheimer reaction. And in Africa, 50% of the people die from this reaction. Okay, so during the process of this whole tick and bat thing, Victoria Campbell from New York State, who sent me the 58 ticks, I believe, said, Jim, I got some weird ticks on this one bat. So look at the ticks, and there's one on the eye. That's a big brown bat. And here's a close-up of the two. They turned out to be Ixodes scapularis. So Ixodes scapularis will feed on just about anything. And the reason why it fed on these bats is because they were injured. They were on the forest floor. Ixodes scapularis is not going to climb up a bat roost. So it was a feeding by opportunity, parasitism by opportunity. And we didn't test these for any pathogens. We don't know what that would, if that would tell us anything. So conclusions. The overall is we created the first formal list of ticks of New Jersey, updates forthcoming. Found no evidence of Lone Star ticks or American dog ticks carrying Rickettsia rickettsii in New Jersey. We were the first to find Carios kellii in New Jersey, and Carios kellii associated with the big brown bat Heptacecus fuscus in New Jersey. That's where I think all the ticks came from, were either from big brown bats or where big brown bats had lived. And we found evidence of Rickettsia and Borrelia in some of these ticks from Carios kellii, and also found evidence of transversal transmission of spotted fever group Rickettsia. And the first record of Ixodes scapularis on the bats. I'm sorry, yeah, on the bat. First shown in North America. Actually in the world. Ixodes ricinus, the European counterpart, has been found on bats, but that's not Ixodes scapularis, and that's not North America. Okay, I'd like to acknowledge my committee, Dina, Andrea, Cesar, Alvaro, and Rich, and my consultants and sometimes drinking buddies. The Bat Tick Network. I might put a shameless plug in to Victoria Campbell and put the information somewhere so you can send them a donation if you want. I'm not forcing you or anything. Risa Pesapani from Ohio State, Cheryl Sullivan from Vermont. She's going to and then family support, everyone here. And this was artwork by Olaf Hajek, and it was from a New York Times Magazine article about spillover events and how we contract zoonoses. There were prejudice. There was no ticks on it. They put a mosquito on it, but no ticks. So I put one up here to make the picture complete. And I think that's it. Here's my visual acknowledgement slide with some familiar characters in there. So thank you very much. Hope you enjoyed it.
Okay, people, we have time.
Greenhouse a few weeks ago.
I have questions.
So all I got was his leg going in. Oh, this is my dog Lucy. She's passed on, but she was helping me collect ticks. That's a squirrel in her mouth. No Ixodes marksi though.
Yeah, there's going to be some echo, so mute yourself there because your computer, you cannot hear.
Okay, I mute this. Speak.
Done?
Mute.
Sorry.
Okay.
Dina, we can't hear you.
I understand, George, that you can't hear me. That's why we're trying to do some like
Oh,
Jim
okay,
was saying,
okay.
shenanigans.
Yeah.
Okay, so now we have the audience. As you can see, we have a beautiful audience in here. You guys should be here. And it's open for questions. So as usual, raise your hand. And those online, raise your hand, and I mean, actually, we can see it there, and we can call up on you, okay? So go for it, ask some questions. The committee, please abstain from asking questions. We're gonna grill them. Okay, any questions? Jim, call on them, okay?
Dana.
I have a question for you, essentially, Mackenzie, also about the backtick behavior that you've seen. So I believe some people don't want bats in their houses, so they'll try to get rid of them. Do you have any experience in what happens to the backticks after that? Like, do they actively migrate towards where other living things are going on?
Mackenzie would have an answer to that, but I think what she's done in the past is, I don't know if they can hear me, is built a bat house nearby that house. And then she evicts them at a certain part of the year when, after the babies have been weaned, et cetera, et cetera. And then she blocks the house off and then maybe puts up a bat house.
Yeah.
Yeah.
Do you have any hypotheses about why Borrelia was so confined?
No,
It was multiple sites close together in Pennsylvania?
they were close within like 20 miles or so, but I'm just gonna guess and say it's probably just the right host they found. But I don't know if that's the answer or not. But yeah, because we had the first batch that were all together. Oh, and by the way, the first batch that were positive were from 2 different bats.
So that was my sort of
Yeah.
follow-up with, do you know how many bats were in those roosts?
Yeah.
You know, are these things transient? Are there 30 bats one night and then 100 bats the next night? Or is it like 2
In the
and 1?
roost?
Yeah,
I think it's steady. However, I should mention that these are from bat rescue people. So they either get injured, people find them, they run into something,
okay.
or they're sick. So I get them from bat rescue people.
Okay, so the PA samples were from bat rescue.
Correct,
folks. Okay.
correct.
Did you say that the Johnsonite, like Borrelia was in larval ticks as well, or were those?
I think, no, I don't think in that study there was Borrelia Johnson in water.
Okay.
I think it was mixed in adults.
Guys, you need to enunciate, otherwise these guys are not
Oh.
gonna hear it. So we have 26 people online.
And they probably heard nothing I just said.
Well, then say it again.
Okay. The question was, were any Borrelia johnsoni found in the larvae? And I think the answer is no. Suggesting it's not transmarial, although, is that not true?
No, I'm saying I didn't realize that.
But-
go ahead.
But you have to remember, some Borrelia, some relapsing fever Borrelia are transmarially transmitted, others are not.
Yeah.
So it's not uniform. What else? Oh, yeah.
You said the Borrelia johnsonite that was found in a human. Has it been found in any other hosts, or is it only ever been found in a bat tick?
As far as I know, it has not been found. And I don't even think it's been found in bats. I think maybe serologically, but I don't,
There's that thesis.
yeah, but there's no, yes, that's right. There was a master's thesis that I found 2 weeks ago from Arizona. She found serological evidence that the bats were infected with Borrelia johnsoni. But the infection, they suppose that the infection is so low that it's just a normal flora, maybe. I don't know if that's the proper term. And she said, you can detect it by qPCR, but when you go to amplify it by conventional PCR to get something to sequence, it doesn't work. So it's in very low abundance, I suspect.
We're following up. This is where we have, remember I asked Jack Foster for the samples and see, maybe we just leave, it would be bad and we'll find very small amounts of DNA. So it did nothing ever was published from that thesis.
No.
Kind of sad. But it was the first time that the bats have nothing really examined.
It was a good thesis, but yeah.
Well, how was it identified in the human that was positive for johnsonite?
PCR, and then probably they PCR like 6 loci, I think. So, and this is done by Tom Schwann, who's like relapsing Borrelia fever got at the Rocky Mountain Labs in Montana.
But this is actually raising, I mean, we're not talking entomology people here, sorry, but this actually raises this question about in a normal host. You were just talking about that in a normal host. I was with Julia Brannan about chiggers. In a normal host, you may not actually get a big infection, so the bats may just keep the infection very low. But you go into a human, like the chiggers, and you get a huge response. You get a big infection and potentially disease. So
Okay.
what you're able to do in a human may not be the same thing you can do in a bat. That's a complicated argument, but that's.
No, that totally makes sense. That's right.
Okay, any questions? Yeah, any questions from the audience? I see somebody,
There's a chat box.
Chelsea.
Chelsea.
21.
Oh, hold on.
Hi, can you guys hear me?
We can
Yeah.
hear you.
Uh,
Don't do that.
Sorry.
All right.
So I have two questions for you, and they will definitely kind of reveal how little I know about ticks. But I found it really interesting that the larvae have 6 legs and then the adults have 8. Is there any?
I was afraid of this question, and I don't know the answer to it.
Oh, okay.
Honest
If
with
it's
you.
okay. All right. So let me go to my next question. And this is another one that you might not have an answer for because an emergency room doctor didn't have an answer for me. But I was wondering, why don't they test to verify these tick-borne diseases?
You mean in a patient?
In patients where they see the rash and they're like, oh, well, you've been bit by a tick, you must have this, here's your medicine and goodbye.
Oh, well, that's a political issue because some physicians, well, first of all, with Lyme disease, if you have a bullseye ring rash, that's almost pathognomonic as a diagnosis, but A, not everyone always gets a rash, and B, it's not always a bullseye. Plus the test is not that accurate. I don't know if that answers your question.
Ah, I see. Okay, yes, thank you very much. And fantastic work. And great
sorry?
presentation. You had a great presentation.
Oh, thank you.
So,
Okay, Dr. Shanglu one.
John Lu.
Hi, yes, hi, Jim. First, thank you for sending me the bat bugs specimens.
Yeah.
I have some comments about how the spotted fever rickettsia transmitted between the ticks, the bats, and the rest of the system. How the bacteria was first, transmitted to the bats in the first place.
Oh, this is a chicken or egg question.
Oh, okay.
Okay, so the easy answer, I think, is it can transmit transstadially because the rickettsia usually are spread throughout the tick and the hemolymph. So it's probably just goes along with the rest of the tissue. How do they contract it in the first place? We don't know, or I don't know.
Does the bacteria has any effect on the bats?
Don't know that either.
Okay, can you, is that possible the bat bugs also get infected with the bacteria?
Yeah, it is. They found serological evidence of immune response to the rickettsia. And Chang Lu, before I forget, I tested 14 of the bat bugs and they were negative for rickettsia.
Oh,
Are they coming from the bat bugs? We don't know. So yeah, did I answer your question?
yes. So yeah, I was curious if the bat bugs also can help transmit the bacteria.
Ah,
But I guess it's not.
okay.
Well, as far as I can tell, yeah.
Yeah.
These are bat bugs taken from the same specimens that you had positive bat ticks from, correct?
No.
Okay, then we don't know.
Sorry.
All right, Zoe.
I had a related question, kind of, Dr. Wong might know this. Do you know if bat bugs use that traumatic insemination.
Yeah, I don't know.
Because that's why they say.
So the question is, do bat bugs use traumatic insemination? And that's what human bed bugs do to inseminate the female. I don't know what kind of shenanigans go on with the bat bug.
I guess yes.
Okay, pathogens, right? Because they have to mount the immune response to
Yeah.
that,
Yeah.
they're refractory to a lot of pathogens. I just didn't know if that was the
That
same way.
that seems like such a just so story.
I have no idea. I heard that here.
Tom?
Yeah, probably a naive question, but I'm just trying to follow the story here. So it sounds like you were implicating the American dog tick in the incidence of Rocky Mountain fever in patients. But it seems that it was very surprising that you didn't see any evidence of it so they're not a host. But at the same time, so you saw a really big prevalence of this pathogen in the bat tick, but you also mentioned that the bats don't feed on anything but bats. So the patients that did show, or that did come down with Rocky Mountain spotted fever, were they bitten by a bat? What interaction did they have with some kind of host where they would come down with this pathogen, or did it not come from the bat
human.
tick? Yes, correct.
Okay,
Yeah.
There's a lot of going back in that question.
So the question is basically, do bat ticks bite people? And yes, they will. In one of those papers from Iowa, the residents of the one home found hundreds of bat ticks in their furniture and on their walls. And they did notice bat bugs. And in another paper, Gill published this in 2005, I think. They discovered one engorged larva or nymph, maybe, whatever, that had human blood in it. So they will bite humans.
And we actually had the Vermont one had dog
Yeah.
blood on it.
The one ticks from, we helped work with, Cheryl Sullivan from Vermont, fed on a dog, a beagle.
Well, we don't know if it was a beagle because the sequence is Julia Brandon actually did an amazing job on that. She's here.
Alex?
I got a follow-up question. Just kind of connecting this like Rickettsia prosecca, where it's transmitted through like last species. Do ticks defecate? And like, is that something
Yeah.
that could be possibly connected to?
So ticks defecate. However, soft ticks have a different mode. They have coxal glands to release fluid. And Tom Schwann has isolated Rickettsia Borrelia johnsoni from the coxal fluid exuded by infected ticks. And a recent publication, as an aside, suggested that soft ticks release coxal fluid to cool the bodies. I don't know if that helps or
The
not.
follow-up would be, would that be infectious?
Yeah, that's how Tom Schwann, well, I guess if you injected it or,
Right.
Okay.
You have to like-
yeah, so,
It's like bacteria.
yeah.
But they're intracellular bacteria. We're talking about the Rickettsia here, right?
No, the Borrelia johnsoni was released by the coxal glands.
I mean, what's the likelihood that you would have a spirochete somehow make it into a cut and somehow make it into the circulation?
Mhm.
And you will die waiting for, that, I don't know, actually, you know. Also,
I don't think they will make it, but I mean, they also in the host they invade the bladder, for example, and they are secreted in urine. But we know direct contact is not a way of transmission.
you gotta have they have to have a sturdy bacteria to do that.
So I don't think it's a dead end for them, although you can find them. But that also tells you about the distribution of the bacteria within the host, because typically relapsing fever are associated with salivary glands, right?
Oh, that's right.
So being excreted in the coxal fluid, that tells you that maybe this Borrelia johnsoni is also invading other tissues in the tick.
Okay, so the question was about where Borrelia johnsoni is in the tick. And I should have mentioned, I forgot to mention, is that, as Alvaro said, the spirochete can be found in the salivary glands of the tick. So this is why they can transmit it under a short time span. So the Lyme spirochete takes 36 to 48 hours to become transmitted, because that's sequestered in the midgut and doesn't become released to the salivary glands for that length of time. So I guess the relapsing fever belly and soft X's are just ready to go if they're already in the salivary glands. Thank you.
Okay, any other questions from the, so it's right now exactly 12 noon, so we have officially finished the